Hello, and welcome to our webinar applying AnteoBind activated Estapor Europium microspheres for development of fluorescent lateral flow immunoassays. My name is Matt Cousins, manager of the Diagnostic Solutions Customer Education and Training Team, a part of the life science business of Merck KGaA, Darmstadt, Germany, and I will be the moderator for this webinar. Before I introduce our speaker for today's webinar, I would like to cover a few items. Please note that today's webinar is being recorded, and we'll be able to share a link with you after the event is complete. We also invite you to submit any questions you may have. Please enter your questions into the question tab located below your video player. You may send your questions at any time during the presentation, and we will address them at the end. At this point, I'm pleased to introduce our speaker for today, Charlie Wang, Head of Diagnostics and Life Science at AnteoTech. Dr. Charlie Wang has more than 20 years of experience in multidiscipline environments. He is a full-spectrum industrial executive with experience ranging from concept generation, upstream research, product design, development, manufacturing to commercialization. He has demonstrated scientific expertise in the fields of life science, in vitro diagnostics, biotechnology, and nanotechnology. He also has significant commercial expertise in business development, commercial strategy, project and team management, technology analysis and transfer, licensing, grants, and intellectual property. Prior to joining AnteoTech, Charlie held senior management roles in several publicly listed healthcare and IVD companies, including PanBio Limited, Alere, Celestis Limited, and QIAGEN. He is currently working with the Anteo team to develop strategies, patents, products, and services for industrial and life science applications and commercialization. So at this time, I would like to turn the presentation over to Charlie. Charlie, the audience is all yours.
Okay, thank you very much. So thanks like Minipro, like Sigma, providing this a great opportunity, allowing us to share a little bit what we have developed in the past several years. So in that, the presentation today I'd like to cover is applying AnteoBind activated Estapor Europium particles to develop a fluorescent lateral flow immunoassay. And I'd like to start my presentation. So the agenda for the webinar today, we are going to have a short introduction of our company, AnteoTech. Then we will discuss the ideal conjugates and protein conjugation options. Then we'll introduction of AnteoBind nanocoating technology and the advantages of AnteoBind activated Estapor Europium microsphere in an immunoassay, and the physical characterizations of the particles after conjugation. For example, is the size, dispersity, and surface charge. Then we'll cover on the performance of AnteoBind activated Europium particles and the conjugates in the functional lateral flow assays, and some discussion on the conjugation conditions. AnteoTech is a company located in Brisbane, Australia. Is a subtropical weather, which is quite welcoming. And we are ISO 9001 and ISO 13485 company, going to become ISO 13485 in July this year. And we have developed a proprietary nanometer binders, primers, and coatings utilizing the metal ion polymers as the base for nanotechnology applications. Currently, we have two main applications for this metal ion polymer technology. One is in energy area, which is mainly focusing on the lithium-ion battery, and the other area is in the life science applications. So currently, we have have products in the commercial AnteoBind product for application is in life science research and development, and also in vitro diagnostic tests. Before going to talking about the AnteoBind, I'd like to go through the lateral flow principles. I believe many of us have been quite familiar with. In the lateral flow, we know that there's certain components, and many components, we may say, from the sample pad to conjugate pad to cover with the membrane, and then have a absorption pad. And it's a composition. In that, we have a test line and control line with antibodies strapped onto it. Actually, one of the important or key components here we can see is the protein particle conjugate. In this protein particle conjugate, it can be controlled really well in size and distribution, and also the functionality of the antibody on it can work really well. And the whole lateral flow assay can perform really well. Though we know that yes, other components may have the kind of applications, and also they can affect and impact on the performance of the lateral flow. But we have noticed that one of the most important factors or components is the conjugates for the lateral flow strips. So in that, we'd like to discuss further on the ideal Europium particle conjugates for lateral flow assay development and for manufacturing. The particles are pre-active and ready to use. So why this one, it will be nice to have because imagine that if the particle is already pre-activated and ready to use, don't need to activate it during each manufacturing batch or each research and screening of antibody or protein process. And it's a very tight particle size distribution and consistent surface charge. Size distribution is very important for lateral flow. We can't control the size after conjugation. You have a mixed population of different sizes. Then to flow through the lateral flow strips is very easily have some inconsistencies. Then the surface charge is also important too, because the conjugate doesn't have a consistent surface charge. The negative charge is the colloidal stability won't be there and easy to aggregate and fall apart afterwards. And also excellent reproducibility, like a batch-to-batch consistency, and easy to scale up for the applications in the lateral flow. That is important, as we know. If we have a failed manufacturing batch, then it will need a lot of troubleshooting and etc. in order to bring the product back. Then can improve the lower limit detection of the assay if possible, because some of the assays are really difficult to go down to certain lower limit detection due to a lot of reasons. And one of it contributed by the particles may be the antibody or the proteins on the particles or microsphere is not active enough, or they lost the activity after conjugation, or the aggregates, etc. And can be utilized for both a qualitative and quantitative tests, and then very similar overall cost of goods to colloidal gold, if it will be nice. And the fluorescent readers are commercially available and can be accessed. So that's the ideal Europium particle conjugation listed here for lateral flow assay development and manufacturing. And to discuss the ideal particles for a lateral flow, and we now have a look at the bio-conjugation options for the protein conjugation. As we know, there are several methods, conventional methods like a passive absorption. If the antibody or proteins, they have a certain good interactions, those are usually hydrophobic interactions, absorption towards the particles, the Europium or other particles, they can be utilized using the passive absorption. It's easy to perform. It's not complicated. It just mix particles with the protein solution together, and the in certain pH and certain additives in it to assist it, and it will be able to achieve some results. The downside of it, as we know, some protein or antibodies just don't bind to those passive absorption particles. Then for the chemical covalent methods, it's a conventional method, we form the chemical bonding covalent bonding through the, for example, like EDC and NHS, that kind of chemistry. But as we know, this conventional chemistry also has its limitation as well. For example, like EDC chemical is very sensitive to moisture, and the batch to batch, they kind of a reproducibility during the conjugation, and also some antibody or proteins are not compatible with this method. And also, we may consider using binding proteins as indirect methods to bind to the antibodies or proteins using a streptavidin biotin interaction. So you coat the streptavidin to the particles, then coming with a biotin-aided antibody or proteins. Also be able to achieve the results what we are looking for. In some areas, people using protein A and protein G as indirect binders. So with one layer of protein A, protein G, or combination, can easily hold the antibodies onto the surface. But this method won't be able to utilize for antigens. And there are several different kinds of binding proteins. Even just direct antibody can also bind to the proteins and as antibody antigen interaction. And the alternatives here, if we have tried the passive, the chemical covalent methods, and all the indirect binding methods, there's one AnteoTech company has developed called metal ion polymers, which is called AnteoBind. It's a new alternative in your toolbox, particularly can assist the bioconjugation process. So what is the AnteoBind technology? The AnteoBind technology for life science, it basically the metal ion polymers. The metal ion polymers is new alternatives. It's a water-based nanobinders being activated. When it's ready to use, it means that it maintains the team solution more than a year. And after activated the solution stability in a year, it means that the store 2 to 8 storage, the particles is stable for a year. It means that it is ready to use. And so it means that after activated Europium particles, the Europium particles can be stored when we need it, we can utilize it as we need. For example, during manufacturing runs, you can activate a big batch of the Europium particles, put it aside, then when you need it, just use a portion you need, for manufacturing runs. Or during the R&D, during the product development early phases in the antibody screening process, we can use the same batch of activated Europium particles for all the experiments is required to perform. So it means the kind of comparison and the checkerboard experiments will be easily can be performed. And the active binding, it means that it's effective and reproducible. As a metal ion polymers is the kind of binding is relies on the gentle multi-point coordination binding to retain the protein activities, and it's reproducible and simple scale up. I have a quick diagram to demonstrate. If it's a carboxylated Europium particles, microsphere surfaces carrying carboxylic acid on the surface, then it will be really strongly negatively charged. And the aqueous metal ion polymers, it's called AnteoBind, comes in to activate. It's simply mix the two solutions together. Then the surfaces will be activated. and it stays activated because the surface becomes very strongly positively charged, and the kind of colloidal stability is a very high, very good. And afterwards, after the surface is activated, then it can be stored for a year and ready to be used to bind to the antibodies or antigens. So then the next is it's called multi-point coordination binding between the metal ion and the antibodies or antigens. So the proteins or antibodies can be gently bonded to the metal ion surfaces and to the particles, and through the metal ion polymers to the particles. So this kind of multi-point coordination binding will provide the kind of quite gentle binding, make antibody or protein quite happy on the surface because the kind of binding is different to the covalent binding. And it is through a multi-point of a weak coordination sometimes, and also additive bonding in that to provide a kind of stability. So the process actually is pretty straightforward. So AnteoBind Estapor Europium particles conjugation workflow, the activated Europium particle conjugation process. So the activation prior to come to this step is roughly an hour. So the Europium particles mix with AnteoBind, and then it's activated, and it can stay in the activation solution with some another treatment. And when it is required to conjugate to the proteins or antibodies, firstly, they wash the activated particles roughly half an hour. And after that, we put the protein and antibody solutions to conjugate to the particles. Then the conjugation step is happening at room temperature with a gentle agitation for an hour. Then after that, during the blocking step, we can block with a DSA or casing or some synthetic blockers of a choice, and you just add directly into it without separating the antibody solution, just add a higher concentration of blockers into the conjugation. Then afterwards, the particle is ready, and then buffer exchange for storage. So the overall the workflow is roughly about 3 hours after we used activated Europium particles to start with. So we can see from here the step is quite a simple particle activation, and during the conjugation and the blocking and the storage, there's quite the hands-on time. It's not that long. It can be finished usually in the workflow. We start with the particles in the morning. Usually in the afternoon, we should have the particles ready to run some half strip assays in the afternoon. So this is the workflow of the conjugation step. Then after the particles is being prepared, or during preparation, there are several characteristics we have mentioned previously is important for the ideal of the Europium particles. And here we give an example using cardiac troponin I antibody conjugation process as an example. So in that you can see that on the left-hand side here for the size measurement. In the size measurement, we mentioned that you need to keep the particle size distribution very tight. The reason you need to keep the size distribution very tight with a very good polydispersity index lower than 0.1, that's ideal, to make sure that the particle size doesn't change too much before and after antibody conjugation. So if we see this kind of a curve about using a laser scattering measurements, shows that the size start to shift too much into like aggregates, then these conjugates cannot be utilized in the lateral flow because the pore sizes of the natural cellulose membrane not allowing it to run properly. So the step is like the carboxylate is Estapor stock particles. The size usually sitting about 320-ish in the like nano size, the zeta size, nanometers. The polydispersity index 0.03, and the potential is here is called a surface charge. The surface like surface zeta potential. Usually because the carry carboxylic acid is in the minus 41, then we're coming with AnteoBind metal ion polymers as to activate the particles, and after activation, the zeta size increase a little bit, and the polydispersity index is very similar, but the zeta potential shift to very positive to 42. And coming with the antibody and the blocking with the BSA, so the anti-CTNI, the conjugate after being the storage, if the PDI is also similar to 0.03, then the zeta potential is a minus 20. So you can see here on the chart on the right hand side, this is the the zeta potential on the surface charge. So in the beginning, it's the bare particles, and the bare particles is in the negative, like zeta potential. So it's a negative. And after the particle is activated, it shifts from very negative to very positive. So this is a very good indication that the particle has been activated successfully. And we can utilize this positive charge of a zeta potential reading as a part of the quality control to ensure that the a particle has a certain characteristics during storage. Then afterwards, it binds the conjugation. The conjugated conjugate will have the surface charge because outside the outer layer will be the antibody and the proteins like BSA as blockers. It carries a negative charge. Then you will come to this the territory of about minus 20, the kind of surface charge. So utilizing this methodology, we have characterized a lot of antibodies or proteins for conjugating to Europium particles, and it gives us a very similar profile for the antibodies we have utilized. And we have noticed that it's very reproducible. It means we do it once, twice, 3 times, or many times, you get nearly exactly the same results on the size and also the surface charge to ensure that we have a very good physical characterization before going down to assay development or production. So this can be utilized as very good reproducible conjugation and in-process QC qualification methods. So until now, particles can be pre-activated and ready to use, provided by the AnteoBind, and also it has a very tight particle size distribution and consistency, and that kind of a surface charge will help to retain the kind of colloidal stability, and very excellent that kind of reproducibility batch to batch. The chart we have presented previously actually can be easily repeated many times and it's seen the same results. Then to the functional assay. So AnteoBind activate Europium particles. This is the next we'd like to discuss. They mentioned about the conjugates, and here on the top, this is the TEM image of the antibody conjugated Europium particles, 300 nanometers, and we are going to focus on the lateral flow assay design principles later on the test line and control line. So this example is the lateral flow assay design. We have like a control line. As we know, we usually have the gold antibody or different species antibody have another particles carrying another antibody as anti-species antibody. And on the test line, we have like an anti-biomarker monoclonal antibody on the particles. And on the nitro cells membrane, we have like another match pair antibody to the same biomarker. Then after being analyzed, come into the lateral flow strips and then flowed into the nitro cells membrane, being captured to form the immunocomplex. Then you have a UV light to excite the particles, and it will generate the kind of fluorescent signal to be detected by the reader. Either will be a scanning reader or the camera-based reader, and to show the images intensity and to compare. So this is the example lateral flow assay design principles. So the CTNR antibody conjugate we have described previously, and we have put into the test using the lateral flow and in the half stripped lateral flow immunoassays just to demonstrate as a model assay. So what we did is we tested it in buffer spike with the CTNR antigens, and in serum spike with the CTNR antigens. And on the left-hand side, here is the results we obtained from the buffer spike with the CTNR antigen. So you can see the signal, and this is the concentration on the x-axis. Very linear. They kind of a dose response between the 0 and 1,000 picograms per ml. We have other results beyond that, but we didn't show it here. Just want to show the lower end of the between a 200 picogram, below 200 picograms. Similarly to the one in the serum, we have the a dose response from 0 to 1,000 picograms per ml. And in this, we have calculated the lower limit of detection in this. So we use about 20 microliter of samples for each a testing point, and each testing point is 3, and we got a very good reproducibility, a percentage of the CV across the doses observed. So the CV is less than 20%, and some of them is less than 10%, 15%. And the estimated lower limit detection, we use a very quick calculation using blank with a 3 standard deviation plus a 3 standard deviation. Is sitting roughly about 20 picograms per ml for both assays in buffers or in serum, which we noticed that, and this kind of observation is significantly better than the colloidal gold-based assay, which is sitting around a 0.2 nanograms per ml, 200 picograms per ml, in the same or similar setting, just to replace the Europium particles with a gold colloid. Then the signal to blank ratio, which also another factor we're looking at, is between the 1,000 picograms and the blank. The signal to noise ratio we observed is more than 50 in both assays. So the result we obtained is quite encouraging. So that's why we kept on testing on different kinds of antibodies and assays. And the next, we'd like to have a quick introduction of the reader we just mentioned. It's important to have a reader. This reader, we collaborate with a company called Assay, and Assay reader is a camera-based reader, which is Australian-based and made. It's a CE mark and FDA ready. It also can accept a wide range of cassettes, and it's a very good software control and offerings and for development, for assay development. So for example, on the right-hand side here, you can see that the test line and control line, if we present it in a way to demonstrating the density or signal intensity after being read by the instruments of the Europium signals. And this is the like a strip images. So the advantages of camera-based the kind of readers is like allows like the visual of the like strips. So sometimes on the strips we may have some like inconsistency of the flow, or may have some kinds of abnormal like kind of lines or dots you can pick up, and then we'll be able to like allowing us to know that it's not the failure of the assay, but it's because of some dots or some abnormal like arranged that kind of a situation is happening on the strips. So that's like what we call the good allowing to have a good strips of visualization, plus a very like compact format for point of care, the kind of settings. So we utilize the reader and collaborate with Accent to like look into the sepsis tests. And as we know, the sepsis test, there are many markers that people are looking at in order to demonstrate it as a model assay. What we did is we developed a like a triplex, the kind of model assay. In this triplex model assays, what we did is utilizing like three like markers. Is a C-reactive protein called CRP, interleukin 6 IL-6, procalcitonin PCT, so the CRP, IL-6, and PCT antibodies. And the design principles we did is like on one particles, we not only just have a one antibody. So what we have tried to demonstrate the capability of AnteoBind metal ion polymers can do is we try to create multifunctional like antibody conjugates. So three antibodies, the CRP, IL-6, and PCT. These three antibodies are pre-mixed in the optimal ratio. It means you can like change the ratio according to the like signal intensity and performance. And in this case, the CRP content amount is really, really low, because the CRP we don't need that kind of a high that kind of a sensitivity. And but the others like a PCT and IL-6, we need to use a higher kind of ratio, and they can conjugate it to the AnteoBind activated Estapor Europium particles simultaneously. And by doing so, it simplifies the conjugation process for the exploration like phase and the purpose for assay development as well. So on the right-hand side here is the like the strip design. The strip design, we have a control line as you mentioned, and then we have a CRP, we have IL-6 and PCT, and we put PCT at the front because that's the marker we'd like to focus on. And but it's up to like when we design it, we'd like to put IL-6 at the front or PCT in the middle. It's like up to our design. And usually put a CRP at the back because we noticed that the CRP signal usually is pretty strong. And quickly short some results we have obtained so far using the Estapor particles activated by the AnteoBind. These are full strips, so it means that it's not just half. We already dry down the prior to this, we have done a lot of half strips, like a single assay work, individual CRP, PCT, and IL-6, or demonstrating very good dose response and the kind of usability. And afterwards, we decided to combine it together as a multiplex assay. So in this multiplex assay, the particles is dried down to the conjugate pad, and we have a sample pad as well. And using the serum, 50% the human serum, spike with like the PCT and IL-6 and CRP, etc., for this kind of purpose. So what you have obtained on the left-hand side here, you can see the x-axis. The x-axis is the PCT antigen concentration, and the y-axis is the signal. And this is the dose response, the curve bar chart, demonstrating that the PCT detection using the Estapor Europium particles activated by AnteoBind will be able to achieve roughly about 50 picograms per ml in the PCT assay. On the right-hand side here is the picture we collected from those strips. So you can see then the background is clean and gradually increase the kind of intensity through the a higher dose of the PCT in the solution. And the result 50 picograms per ml, which is we according to the literature, is roughly sitting about at around 500 picograms per ml using colloidal gold based assays, single assays. And the next one we'd like to share is the IL-6 result. In this proof of concept, the sepsis marker, IL-6 is in the middle of the three tested together. This one, because this serum already has a quite high CRP, so we utilize this serum to see the CRP, high concentration CRP will be able to affect the IL-6, and it doesn't. And in this way, we have on the x-axis is the IL-6 antigen concentration, the y-axis is the signal, and there's a bar chart to demonstrating the dose response of the IL-6 antigen. And roughly, we calculated the estimated lower limit of detection is sitting around 10 to 20 picograms per ml for IL-6 assays, which is the same, significantly better than published in the literature, about 200 picograms per ml. Similarly, for the CRP assay, and the CRP assay, I think we have a very sensitive CRP test, which can easily go down to 50 picograms per ml. So in the future, when we need to develop into real sepsis test, it means that we need to reduce the sensitivity in order to fit into the clinical range. So on top of this, both CRP and the PCT and IL-6 and PCT, both assays actually fit into the potentially the sepsis clinical range and cutoff. So afterwards, it's a triplex assays. We have achieved the kind of proof of concept in the full strip format for the sepsis detection using the Europium particles approach. So above is some functional assay using AnteoBind activate Europium particles technology, and so far we have done not only just these assays, but also other model assays in our hands, demonstrating similar performance. We have observed in different point of care lateral flow application areas, so including the infectious disease as well. So the ideal Europium particle conjugation for lateral flow assay development and manufacturers would be nice. We mentioned it would be able to improve the performance, the lower limit detection of the assay, and at the same time can be used both for qualitative and quantitative, similar cost of goods, and also fluorescent readers are commercially available. And here we'd like to share a table with the AnteoBind Estapor Europium conjugation assay, like a component guide here, like in the items. For example, like antibody antigen amount for conjugation and usage, roughly sitting between 100 and 200 micrograms per milligrams of particles. So during conjugation and blocking, we'll have a much higher concentration of the proteins to come into usually at 1% of the protein, the BSA or casein. For separation, there's certain separation like steps. For example, like you want to separate the solutions. Currently, it's because it's small, like in a small preparation using centrifugation. The centrifugal speed is roughly sitting 10,000 RCF for 10 to 15 minutes, depends on the volume. The protein conjugation step, there's several things need to be like to avoid. For example, like the metal chelators, as we are using the metal ion polymers on the surface to avoid using phosphate, EDTA, or other like metal chelators in the like conjugation like steps. And the brokers we have mentioned, we can utilize the BSA, casein, or other like synthetic brokers, commercial available brokers, which can interact with the like the metal ion polymers and the like particle surface. The storage in solution, we avoid using like a high concentration of proteins and those same like metal chelators like phosphate and EDTA. And the drying buffer for conjugation pad, we use some sugar and some small amount of a surfactant and some like small amount of the like BSA, etc. It depends on the like the manufacturer's like requirements. And the conjugate pad pretreatment using like a sugar or small amount of surfactants and a little bit higher amount of BSA just to block like the like conjugate pad. The lateral flow running buffer suggested to utilize treat buffer based with like as the starting point of a surfactant is 1% of a twin 20 and a lower amount of a BSA. If it needs like to like have a lower background, you can increase the BSA and some other surfactants as well rather than twin 20 and some sugar or PEG, etc. to if the viscosity is required to be like higher. To dispense the like to disperse the like particles, we use a fast sonication. The normal like benchtop fast sonication will be able to do the job. But sometimes during like a conjugation, some particles may temporary like aggregation. When the particle is aggregated in this situation, can be easily like separated and dispersed by using the fast sonication. So usually this can be done after adding the antibody to the particles. You can like have a quick fast sonication to provide much better that kind of distribution. The natural cellulose membrane we utilize in the range for the 300 nanometer of Estapor Europium particles is roughly sitting at 90 to 120 HF, 90 to 120. These kind of pore sizes. We have a try higher like numbers because the slower flow rate, but because pore size when it gets too small and it's not easy for the big sizes like Europium particles to flow. So and the conjugate pad utilize the mini pore conjugate and the sample pad. And lastly, before we conclude the presentation, we'd like to talk about the initial scale-up study, and the workflow as the example. So the workflow is like similarly to what we have described, but this time if you want to scale up to certain like a bigger size, like 100 milligrams, for example, they kind of amount for Europium particles. So you prepare the antibody or antigen solutions. It's a mechanical mixing. For example, you stir bar magnetic mixing. Then you add the particle slowly into the solution. Then you'll be coated and conjugated, leave it in room temperature for an hour. Then after an hour, you come back to add the broker solution, high concentration. Then you the broking step is also room temperature for an hour with gentle agitation, or the same, it's like a mechanical mixing in the solution. And the separation and the buffer exchange for storage, usually it's a centrifugal step at room temperature of 2 to 8 degrees if the centrifuge has the cooling functionality. And after wash into the storage buffer, the conjugate can be stored in the storage buffer and ready to be utilized to stripe onto the conjugate pad. And we have noticed that some antibody can be really stable during this step, up to weeks, but some protein is not as stable. It means some storage buffer optimization is required by the developers and manufacturers. And the conjugate in this initial scale up can easily enough for about 20,000 tests per small batch, and it can put the batches together, can easily up to 100,000 tests. Also, this depends on the particle amount per strips as well, and the reader capability. And so the larger volume is also scalable just by changing the vessels if it's required. So this initial scale up study, I'd like to demonstrate here.